Hello guys and welcome back to Excel Academy. So again I'm here with the another lecture. So in the previous lecture we talked about the nucleosome movements like sliding, ejection or the replacement, and in this we are going to talk about the nucleosome positioning. इसका मतलब क्या कि किस तरह से with the help of the DNA binding proteins आप nucleosome को एक fixed position में आप उसको कर देते हैं, ठीक है? So this is done how? That's what we are going to study now. Okay, so some nucleosomes are found in specific positions, and these are really important because those are actually sometimes non-specific, but they are fixed. ठीक है? So ये भी जरूरी हो जाता है because certain regions of the DNA are meant for certain kinds of gene expression. तो उस टाइम पे ये बहुत important हो जाते हैं. ठीक है? So because of their sequence non-specific and dynamic interactions with the DNA, most nucleosomes are not fixed in their locations, and these are really important. Why? See, अगर वो fix होंगे तो काफी gene expressions आपके जैसे for example transcription जो है, replication है, वो नहीं हो पाएगा, ठीक है? So that's why it is important that all the nucleosomes are not fixed. Now, positioning a nucleosome allows the DNA binding site for the regulatory protein to remain in the accessible linker DNA region. Now, in many cases, nucleosome-free regions are larger to allow the binding sites for the multiple regulatory proteins to remain accessible. Okay? So, for example, the upstream of the active transcription sites are frequently available for the it is available or it is associated with the large nucleosome-free regions. Because आपको मालूम है कि transcription में भी, anyways, I'm going to take this in the upcoming lectures. So we need so many regulatory sequences in order to start the transcription side. So उसके लिए भी आपको डीएनए एक्सेसिबल चाहिए. इसका मतलब क्या, वहां पे न्यूक्लियोसोम्स नहीं होना चाहिए. So nucleosome positioning can be directed by DNA binding proteins or with the help of the particular DNA sequence. ठीक है, तो यहाँ पे हम लोग दो चीज पढ़ने जा रहे हैं कि nucleosome positioning कैसे की जा सकती है. So there are two ways. That is, first one is the DNA binding proteins. So the many proteins cannot bind to DNA within the nucleosome. So binding of a protein to the DNA, it can prevent the subsequent association of the core histones with that stretch of DNA. इसका मतलब क्या हो रहा है? And guys, it's very simple. See, you have got the DNA. Now, suppose a protein comes and binds over here. Now, the thing is that in this region, your nucleosome cannot come and form. ठीक है, तो ये एरिया आपका फ्री हो जाता है. आपका न्यूक्लियोसोम भले यहाँ पे हो या फिर यहाँ पे हो, बट दिस रीजन बिकम्स न्यूक्लियोसोम फ्री. So this is meant for the other gene expression. So that is, it is preventing the subsequent association of the core histones with that stretch of the DNA. So it means this stretch of DNA is now free. So if two such DNA binding proteins are bound to sites closer than the minimal region of the DNA required to assemble nucleosome, the DNA between the proteins will remain nucleosome free. So what does it mean? It means that you have got one protein binding over here, one protein binding over here. So this region is actually not available for the nucleosome to wrap around. Okay, let's see the figure over here. Yeah, so we have got certain figure. So you can see that if suppose two proteins are there, they go and bind. So what happens in this region? It becomes the nucleosome free region, right? So what will happen here? So many DNA binding proteins are there which is coming, okay, and they go and bind. So what happens? Since we know that the nucleosome requires at least 147 base pair, now if it is not available, so nucleosome cannot assemble in that particular region. So this region will remain nucleosome free. Okay. Now binding of the additional proteins to the adjacent DNA can further increase the size of the nucleosome free region. तो यानी कि अगर यहाँ पे एक और प्रोटीन ऐड होता है तो जिसकी वजह से क्या होगा, this length is going to be increase. So some DNA binding proteins interact tightly with the adjacent nucleosomes, leading to nucleosomes preferentially assembling immediately adjacent to these proteins. Okay? So this is the second case. You can see in the second case is that, so you have got a subset of DNA binding proteins. Now this has the ability to bind to the nucleosome. So इसने क्या किया है, पूरे एक nucleosome को ही bound कर लिया है. So once it is bound to such nucleosome DNA, now these proteins will facilitate the assembly of nucleosome immediately adjacent to the proteins binding डीएनए बाइंडिंग साइट. सो इसका क्या मतलब हो रहा है कि आपने इसको पोजीशन किया है. दिस पर्टिकुलर डीएनए बाइंडिंग प्रोटीन गोज इन बाइंड्स द डीएनए एंड इमीडिएटली टू दिस पोर्शन ओनली योर न्यूक्लियोसोम विल बी फॉर्म्ड. इसका मतलब क्या हुआ, आपने उस पोजीशन में न्यूक्लियोसोम को फिक्स कर दिया है. ठीक है, सो नाउ आई नो द रीजंस और आई नो द सीक्वेंस वेयर इट कैन एक्चुअली गो एंड फाइंड. Now, depending on the DNA sequences, So now when DNA is bound in the nucleosome, it actually gets bent. Okay, so obviously because it has to make another round around the nucleosome, so it will actually bend. So AT-rich DNA has a intrinsic tendency to bend towards the minor groove. So you can see that it is the minor groove which is making the interactions with the histone octamer, and this minor groove is rich in the AT base pairing. Now, AT-rich DNA is favored in positions, and that's why it is facing the histone octamer, whereas you have got the GC-rich in the opposite. So therefore, it is favored when the minor groove is facing away from the histone octamer. So guys, what you have to remember over here is that the minor groove, which is rich in the AT, is actually facing the histone octamer, whereas the opposite, that is GC-rich minor grooves, are facing the outside, that is away from the histone octamer. So this is just a short lecture, and it was specifically made so that you guys can understand about the nucleosome positioning. So please guys watch continuously so that you can get the idea what exactly we are talking about in the chapter 8 of the Watson's lecture summary series. So stay tuned with the Excel Academy. If you like it, please share with your friend circle also so that they can also get the benefit out of these lectures. Thank you.